I would like to share my research project, which is the characterization of Taman oil, in which I have particularly looked at the fatty acid content, the level of heavy metal, and the microbial contamination in Taman oil, which is locally produced in the Maldives. So without any delays, I'll begin my presentation. First of all, a bit of an overlook on Taman oil. So the Calophyllum inophyllum plant, known more commonly as the Indian laurel or Taman, are huge evergreen plants which are valuable source of lumber of premium quality and seeds with immense medicinal properties. Though all parts of the plant, such as the leaves, barks, roots, all have been used for various purposes, the oil extracted from the seeds of the plant makes it in particular a highlight both commercially and medically. It is considered native to tropical Asia, with the plants being distributed widely across all over Australia and East Africa. And in traditional herbal medicine, the oil from the seed is used to relieve pain caused by cold sores, dermatological problems such as acne, eczema, athlete's foot, and sunburns. And literature reports indicate that the oil extracted from the seed of Calophyllum inophyllum inhibited ocular irritations caused by the DNA damage induced by ultraviolet radiation. And apart from this, the antibacterial activity of taman oil also was studied by testing the neoflavonoid constituents, which were in particular effective against the bacteria which is involved in skin infections, namely aerobic gram-positive bacteria strains of like some of the very common ones that we all must have heard is like Staphylococcus aureus or Staph, and Propionobacterium and Staphylococcus epidermidis. They were all susceptible to the oil within the minimum inhibitory concentrations, which was similar or lower than that of ofloxacin, which is an actual antibiotic. And although the seed contains 40 to 60% of the fixed oil by weight, the generation of environmental pollution as the presence of heavy metals such as copper in the seed is it could be a problem that could arise due to negligent disposal of the seeds. So now, looking at the problem statement, in this day and age where like traditional medicines are competing head to head with the conventional counterparts, we can see it from the current status of the COVID-19. Like there are so many people who still opt for their traditional or ethno-medical medicines when it comes to various medical conditions. So the conventional traditional medicine is like competing head to head with their conventional counterparts and making their way into the mainstream medicine. So it would be of particularly of great economic boost for a country to have their own traditional medicine, right? So Maldives is a country where people have been practicing the use of traditional medicine, or commonly known as Divehi base in our local language, since the ancient times. So like many other countries, the practice of utilizing traditional medicine is still significantly prevalent in the Maldivian society, but due to the scarcity of resources and materials required to compile the actual medicine, the number of locally produced Divehi base or the traditional medicine remains very low. Divehi base comes in all forms. It could be paste, it could be beverages, it could come in tablets, oils, or ointments, or any of the forms. So as far as the subject of discussion, the taman oil is concerned, the production and distribution of it is highly applicable to Maldives, since the Calophyllum inophyllum plants are abundant in Maldives and extracted from the seeds of the fruit of the plant in the Maldives, particularly in Nayani Fuamola. In like the oil remains strikingly popular, mostly due to the fact that almost all of the oils are topical and are made from raw materials assembled from plants in the Maldives. And taman oil is one such locally produced topical oil, which is primarily used to treat dermatological conditions. So, though the oil is able to treat the conditions, the constituents which facilitate the healing process still remains a subject which needs to be explored. So, characterizing taman oil produced from the local plants of the Maldives would be quite a breakthrough in that exploration. So, moving on to the aims and objectives, the aim of this study was to characterize taman oil produced in the Maldives, and the objective of this study was to analyze the fatty acid profile of the oil extracted from Calophyllum inophyllum seeds, and also to analyze the heavy metal profile along with the microbial content of the taman oil. So next, the hypothesis that we formulated was taman oil has medicinally useful fatty acids which are free from heavy metals and microbes. So in the next slide, I'll be talking about the methodology and the procedure that was followed in the extraction of Taman oil. And I'm sure you can see a very scenic like pictures from the left side of the slide. So this is like the island Naviani formula of Maldives, which is like one of the most southernmost islands in the Maldives. So this is the place where the oil extraction actually is carried out. So the inoculum seed oil extraction was carried out by a local who resides in Navina Formula, and the procedure followed in the oil extraction began by the collection of inoculum fruits from the littoral zone of the island, which is the area where the plants are abundant. Then the fruits are harvested and kept in shade for 3 months, after which the kernel inside the seeds were removed and washed. And thereafter, the kernels were cut into small pieces and dehydrated. Now here we use there were the local healer gave us suggestions on two ways of drying the kernels. So there was it could be either by direct sunlight or by it could we could also dehydrate it by using an oven. So in the former method of drying, the temperature is kept at 50 degrees Celsius, and collectively in both the methods of dehydration, the kernels are dried till the total weight of the yields drop down to 2.5 kg of the actual number that we started, which was 3 kgs. So next, the dried kernels were subjected to a cold press machine with a single inlet, where which is the part through which the seeds are fed into the machine. And ultimately, the desired product, which is the taman oil, of course, flows out through one of the exits, and the non-oiled cake residue is collected via the other exit. Next, looking at the laboratory analysis, the collected taman oil was then transferred into sterile bottles and labeled and kept below 25 degrees Celsius until it was sent to SGS Lanka Private Limited, which is a laboratory where we send the samples for analysis. So the samples were collected from Maldives National University by SGC laboratory staff, and the chemical profile of taman oil was investigated through various analytical techniques, which included gas chromatography, atomic absorption spectroscopy, and microwave digestion. Also, in the microbe analysis, aerobic plate count and the presence of coliform were tested by horizontal method of enumeration of microorganisms that are able to form colonies in a solid medium and incubated at 30 degrees Celsius. And pH was also measured, and a pH meter was used to analyze this variable, and water activity was determined through basic principles of determining water activity. So next, I would be looking at the results that we got from the laboratory analysis. So in here, the first table is like we have recorded the data regarding the fatty acid profile. In the data recorded for the fatty acid profile, polyunsaturated fatty acids were found to be the major component, which had a value of 37.31 grams per 100 gram of the oil, followed by the monounsaturated fatty acids with 31.69 grams, and saturated fatty acids by 31 grams. Then there was linoleic acid was the dominating when looking at the individual fatty acids that contributed to the uh, lipid profile. It was said that linoleic acid was the dominating fatty acid with a quantity of 36.88 grams per 100 gram, followed by oleic acid by 31.33 grams, and palmitic acid with 16.58, and at last stearic acid with 12.61. These were the four major saturates that were identified, and minor amounts of other fatty acids such as arachidic, linolenic, and arachidonic acids were also detected in the fatty acid profile. So the overall unsaturated fatty acids were relatively high, since linoleic acid is a polyunsaturated fatty acid and it was present in the greatest amount. And the same thing goes for oleic acid, which is a monounsaturated fatty acid. Now, both stearic and palmitic acid are saturated fatty acids, so it added to the overall quantity of saturated fatty acids. Hence, the trends which are observed in the general categories of the fatty acids were quite consistent with the type of individual fatty acid which were present in the oil. Now, talking about the four fatty acids that were observed in the greatest amount, it could be said that they are the ones actually facilitating the overall healing of the skin elements. We do know that taman oil has some beneficial effects to the skin, but we don't exactly know what are the constituents which are promoting that. So, for instance, according to literature, linoleic acid is an omega-6 fatty acid, which is an essential building block for ceramides, one of the skin's main moisturizing elements. Our bodies can't make this essential fatty acid, so we need to get it from our food, or we need to put it directly on the skin. So since taman oil is a topical oil, it enhances this, or it gets the job done. And linoleic acid helps make the skin barrier stronger, so it can effectively keep water in and irritants out. And linoleic acid is also an anti-inflammatory and helps stimulate cell regeneration. So the topical use of linoleic acid further helps to reduce acne breakouts. So mostly during pubescent and prepubescent adults tend to like suffer from pimple formation and acne. So this oil could be particularly helpful for them as well. Now, likewise, oleic acid comprises antioxidant compounds that help fight free radical damage caused by environmental aggressors like UV rays. We know during our daily hectic life, we need to go out in the sun a lot. And if you're enjoying on the beach, also sometimes, most of the times, you would get sunburned. So this oil helps to fight against these type of aggressors like UV rays. So, and also UV rays are top trigger of premature aging. So, oleic acid present in the oil is in particular helpful for this. And now palmitic acid and stearic acid act as emollients or moisturizers. Emollients are often used to help manage like and itchy and scaly skin conditions such as eczema, psoriasis, and ichthyosis. And these emollients are mostly the ones which keep the skin soft and moisturized. So I'm sure all the ladies, beautiful ladies in this conference, would be really excited by this news. Now, moving on to the heavy metal profile, heavy metals which are toxic at low concentrations, such as mercury, cadmium, arsenic, and chromium, were not detected at all in the oil. However, copper was detected, which was, although it was detected, it was at an extremely low amount. Nonetheless, copper, which is essential to maintain the metabolism of the human body in the amounts it was detected in the oil, hence it can be concluded as not posing a risk of as such. And in my right side you can see the table for the microbe profile, and it shows an absence of coliforms and a significantly low aerobic plate count, which indicates the microbe inhibitory effects of the oil, as well as a low water activity value of 0.62 indicates that the mold and microbial growth would be inhibited. This would result in a longer shelf life, and this is an important property since it is used to predict the stability and safety of the food with respect to microbial growth rates and deteriorative reactions. Um, Now from this slide, you will be able to see the results of fatty acid profiles from previous studies which were carried out on taman oil. A similar trend could be observed in all three of the tables, with the oleic acid being the major constituent, followed by linoleic, then stearic, and then palmitic acid. The results obtained in my study complied relatively well with the data regarding the fatty acid composition of the oil reported in literatures in terms of the values. However, the order of arrangement of the maximum concentration varied in the position of linoleic and stearic acid, because in my study the oil contained the greatest number of linoleic acid, but from the literatures it is they are agreeing on the fact that oleic acid was obtained in the greatest quantity. So although there was a slight difference in that, mostly the values that were obtained were in close relation to each other. And now to conclude, I would like to say various studies confirmed skin active effects of taman oil treatment and antimicrobial protection, which were provided by various constituents present in it. Now that we have discussed about all the fatty acids and stuff, it gives a clear picture of what exactly might be helping in the skin healing. Likewise, from the results of my study, also the results fatty acid profile of taman oil shed light on the therapeutic effects which facilitate dermatological healing. Results of the heavy metal and microbial profile indicates that stability and safety of the oil. Though copper was present in the oil, it was not at concerning levels. Hence, the study findings could be used to promote the prediction of promote the production of taman oil in Maldives as a marketable, authentic local product, and can be produced on a large scale as a safe and environmental friendly as well as an economically valuable products. So that's the end of my presentation, and I would like to thank my supervisor, Dr. Naila, for giving me this opportunity to share my research with all of the participants in this conference. Thank you very much.